Hi, in this video, we are discussing advanced glycation and products, which are products of post-translational modification of protein. Advanced glycation and products, or AGEs, are formed due to degradative post-translational modifications. These modifications are degradative because, as the name implies, they disrupt the normal function of proteins and lipids. The glycation reaction, or non-enzymatic glycosylation, is responsible for the formation of AGEs. This reaction is between a sugar molecule and an amino acid residue on a protein, or a lipid, to form a glycated product. This reaction does not require any enzyme or energy in the form of ATP. Since AGEs alter the normal function of proteins and lipids, they are thought to be the cause of a variety of degenerative diseases such as diabetes, atherosclerosis, cancer, and Alzheimer's disease. As such, there is a strong motivation to investigate AGEs. So first, let's examine how AGEs are formed. There are four main pathways on AGE formation. The first one is the Maillard reaction, which is highlighted by the orange box. The starting point of the formation of AGEs is when the carbonyl group of a sugar molecule reacts with the amine group of an amino acid residue to give a Schiff base, which is characterized by the C-N double bond. In this reaction, water is released as a byproduct, which means that this reaction is an elimination reaction as well. Next, the Schiff base undergoes rearrangement to form an open-chain Amadori product, which then undergoes cyclization, which is a more stable form of the product. Several irreversible reactions occur, such as dehydration, oxidation, and fragmentation, to form AGEs and other byproducts, which include glyoxal, another intermediate for the formation of AGEs. As we have seen, AGEs are formed during the late stages of the Maillard reaction in food and inside cells. The Maillard reaction is involved in cooking whenever you toast, fry, bake, or roast, as long as you get that brown color. The second pathway involved in the formation of AGEs is the polyol pathway. The polyol pathway is a reaction that converts glucose to sorbitol, then to fructose. Fructose undergoes several reactions to form AGEs after reacting with proteins. The last two pathways in the formation of AGEs are glycolysis and the pentose phosphate pathway. Glycolysis is a 10-step catabolic reaction in the cytoplasm that converts glucose into pyruvate. One of its intermediates is glyceraldehyde 3-phosphate. The pentose phosphate pathway also generates glyceraldehyde 3-phosphate as an intermediate. Then, a phosphatase cleaves off the phosphate group so that glyceraldehyde is generated. Glyceraldehyde can then react with amine groups of amino acids to form AGEs. Food is one of the main sources of AGEs, and the cooking method affects the amount of AGEs present in food. High temperature and low moisture result in higher AGE content, as this increases the chance of burning and charring food. Fried, charred, and roasted foods have the highest AGE content. Common AGEs include carboxymethyllysine, or CML, and argyroperidine. The epsilon amino group of lysine is modified to form CML, while methyl glyoxal is covalently bonded to arginine to form argyroperidine. These AGEs exert their effect when they bind to their receptors, known as receptor for AGEs, or RAGEs. RAGEs play an important role for the normal physiology of our body. Once the ligand binds to the receptor, a signal transduction pathway is initiated that will eventually result in the transcription or silencing of genes depending on the environment. This leads to the expression or downregulation of proteins to respond to the environmental conditions. For instance, RAGEs have been implicated in the normal functioning of our lungs. They are involved in the formation of alveoli, which are tiny air sacs that are the sites of gas exchange. Moreover, RAGEs are important for the lungs to stretch and expand when we inhale. RAGEs are also crucial in the strengthening of our bones by increasing the mineral density and biomechanical strength. RAGEs are also important to mount an immune response against pathogens and contaminants. Lastly, RAGEs are important for the differentiation of cells in the nervous system into neurons, glia, and other specialized cells. These receptors also trigger the migration of neurons from one part of the brain to the other, and they are also crucial for nerve regeneration whenever there is nerve damage due to injury and trauma. There must be a balance in the amount of AGEs so that homeostasis is achieved. Thus, we need a system to clear excess and unwanted AGEs. Intracellular AGEs are degraded via the action of proteases. Then, they are released into blood plasma where it is filtered into the kidneys. AGEs are then excreted in the urine. However, proteins found in the extracellular matrix are more resistant to proteolysis. Thus, AGEs found in the ECM are less likely to be degraded and excreted. When there is an excess of AGEs, symptoms of aging start to appear. An abundance of AGEs result in abnormal functioning of cells and tissues, which leads to diseases that are related to aging. There are 5 main effects of having excess AGEs. There is an increased concentration of toxic reactive oxygen species, an increased risk of cancer and metastasis, an increase in the release of pro-inflammatory cytokines, damage to the extracellular matrix, and an increased risk of cardiovascular diseases. Reactive oxygen species, or ROS, are produced as byproducts during the intracellular formation of AGEs. ROS is toxic to the cell and has been found to induce mutations to mitochondrial DNA. When this occurs, defective mitochondrial proteins are synthesized and mitochondria stop functioning normally. This triggers the apoptotic pathway, and when a large population of cells die, aging-related diseases begin to manifest themselves. Moreover, AGEs have been implicated to increase the risk of cancer and metastasis. AGEs bind to the receptors to induce a mitogen-activating protein kinase, or MAPK, signaling cascade. Downstream of the signaling pathway, the transcription factor NF-kappa B is activated, which binds to several transcription binding sites in our genome. NF-kappa B enhances the transcription of the gene coding for vascular endothelial growth factor, or VEGF. Thus, there is increased number of VEGF proteins. VEGF has been shown to drive metastasis as this protein is important for the formation of blood vessels. Also, AGEs affect our DNA. Histones are rich in lysine and are often glycated to form CML. The normal packaging of DNA is disrupted, and DNA is no longer tightly bound to histones with glycated lysine. Thus, DNA is more exposed to several transcription factors, which can result in the synthesis of unwanted proteins. Moreover, DNA that is not tightly bound to histones are more susceptible to carcinogens, leading to an increased risk of mutations to our genes. This can drive the progression of cancer if the cells have acquired the necessary mutations for them to become cancerous. In addition, AGEs have been shown to affect the immune system. An excess of AGEs have been implicated to stimulate macrophages and endothelial cells to release pro-inflammatory cytokines such as tumor necrosis factor alpha or TNF alpha and interleukin 1 or IL-1. TNF alpha in particular has been shown to form thrombus or blood clots in blood vessels. These pro-inflammatory cytokines trigger vasoconstriction as well, leading to the decrease in the diameter of blood vessels and hence atherosclerosis. Also, AGEs increase the amount of ECM proteins. This is because there is an increased production of tumor growth factor beta or TGF-beta, which binds to its extracellular receptor. This triggers the transcription of genes that encode for proteins found on the extracellular matrix or ECM, such as collagen, laminin, and fibronectin. AGEs then bind to these ECM proteins, resulting in decreased strength and flexibility of the ECM. AGEs also interfere with interactions between cells and the matrix. Furthermore, AGEs lead to vascular stiffness when AGEs bind to elastin, a common ECM protein that imparts elasticity. This causes blood vessels to be stiffer, which means that they contract and expand less, leading to increased strain on the walls of blood vessels as well as high blood pressure. AGEs stimulate endothelial cells to release endothelin 1 and downregulate the production of nitric oxide. Endothelin 1 is responsible for vasoconstriction, while nitric oxide plays a role in vasodilation. Since there is an increased amount of endothelin 1 and a lowered concentration of nitric oxide, the diameter of blood vessels decreases, which can eventually lead to atherosclerosis and high blood pressure.
From our literature review, we have found many diseases which are related to AGEs. One common illness is diabetes, which is characterized by high blood glucose concentration. The production of advanced glycation end products is significant in aging-related diseases. This is because the production of highly reactive aldehydes is elevated due to continuous hyperglycemic status associated with oxidative stress, inflammation, and disorders of glucose metabolism. This results in the rapid formation of AGEs from proteins in a short amount of time. Furthermore, AGEs can also directly result in modifications in the insulin hormone, as serum proteins are frequent targets of modifications by sugars and reactive carbonyl compounds. Hence, the insulin is less recognized by the cells, and this results in insulin resistance as they are less sensitive to insulin. As explained earlier, the increase in expression of TNF alpha also results in the increase in vascular stiffness and hence diabetic arteriosclerosis. As a summary, we have three main physiological mechanisms that lead to arteriosclerosis. The first one is diabetes, which leads to an increase in vascular stiffness. The walls of arteries become inelastic and are unable to contract and expand as much as before. The second one is vasoconstriction, which leads to the decrease in diameter of blood vessels, causing the heart to pump harder, which leads to high blood pressure. The last mechanism is the release of pro-inflammatory cytokines, where inflammation causes the blood vessel diameter to decrease. Arteriosclerosis can be prevented and treated, but first the condition must be carefully diagnosed. There are two main methods to detect for the presence of AGEs. The first one is through an immunochemical method called enzyme-linked immunoabsorbance assay, or ELISA for short. Cells are first incubated with AGE-specific polyclonal antibodies. These antibodies will bind to their specific AGEs. Then, anti-AGE antibodies that have unique fluorescent tags are added. Thereafter, the cells are visualized under a fluorescent microscope. Different colors correspond to different AGEs. The second method that can be used would be through the use of liquid chromatography coupled to a tandem mass spectrometry that performs MS-MS analysis. Proteins are first separated into components via liquid chromatography based on their unique physical properties. Then, these proteins are passed through an MS-MS analyzer, where they can then be identified based on their MS and MS-MS spectra. There are several therapies available that can mitigate the effects of increased AGEs in the body. Firstly, amidoguanidine is a potent AGE inhibitor that traps the aldehyde groups of reducing sugars so that they can no longer be bonded to proteins. This reduces the likelihood of the formation of AGEs. Thus, the arterial elasticity increases and the vascular AGE concentration decreases. Secondly, citric acid decreases the levels of ketone bodies that can be converted to sugars and react with proteins to form AGEs. Lastly, lifestyle changes involving avoiding food that are fried, boiled, or roasted can also significantly decrease the amount of AGEs in the body. We have conducted an experiment to investigate which amino acids are modified in structural proteins that result in heart diseases. Our hypothesis is that AGEs and degradative modification of proteins involved in the structural integrity of cells and tissues can lead to cardiovascular diseases. In order to test for our hypothesis, we used an animal model for cardiovascular diseases. We were given a plasma sample from the aorta of a pig which had ischemic heart disease. The proteins found in these samples were purified and some samples were collected for BCA assay to quantify the amount of protein present in plasma. After purifying the protein, we did desalting to prepare for high pH, high performance liquid chromatography, followed by LC-MS-MS to separate individual proteins into smaller peptides. Finally, database search of the proteins of the aorta samples was conducted. Possible modifications of the proteins identified were also examined. The BCA assay serves to quantify the amount of protein present in the sample by adding BCA reagent to the sample's N standards, and thereafter determine the absorbance of these samples using spectrometry. The results of the assay are shown in this slide, where BCA sample 1 has a higher concentration of proteins than BCA2. The former has a concentration of 9.39 micrograms per microliter, with a standard error of measurement of 1.32, as compared to the latter sample, which has a concentration of 8.62 micrograms per microliter with a standard error of measurement of 0.71. This result is expected because the BCA2 sample has been processed via several rounds of centrifugation and hence some of the protein could have been lost during the process, resulting in the lower concentration observed in the BCA2 sample. The following slides will show the results obtained from performing database analysis in Mascot to determine the identity of proteins from MS data. The fixed modification entered into the data search would be the addition of a carbamidomethyl group at the C terminus. Other variable modifications like the addition of arch-pyrimidine, carboxyethyl, and carboxymethyl groups at the lysine residues were also entered. The data was downloaded and run through Mascot. Thereafter, the results were filtered for highest peptide score and appropriate peptide variable modifications. An expectation value that is lower than 0.05 indicates a good and reliable positive result. Both amino acids are the ones that have been glycated. In this slide, it shows that aorta sample 1 contains the protein collagen type 6 alpha 3 chain from the gene COL6A3. Variable modifications of this protein involves the oxidation at the methionine residue as well as the addition of an arch-pyrimidine group at the lysine residue. A similar method of database search was applied for the second aorta sample. The data was filtered for highest peptide score and appropriate peptide variable modifications to attain the results. This slide shows the results obtained for aorta sample 2, which contains myosin from the gene MYH11. Modifications to the peptide include carboxyethylation, deamination, and oxidation. From the database search, it was found that the first file contained data for protein that is encoded by the gene COL6A3. This gene codes for the alpha 3 chain, one of the 3 alpha chains of type 6 collagen, a beaded filament collagen found in most connective tissue. The alpha 3 chain of type 6 collagen is much larger than the alpha 1 and alpha 2 chains. This difference in size is largely due to an increase in the number of subdomains that are found in the amino-terminal globular domain of all the alpha chains. These domains have been shown to bind extracellular matrix proteins, an interaction that explains the importance of this collagen protein in organizing matrix components. It is also functioning as a serine-type endopeptidase inhibitor. It stops, prevents, or reduces the activity of these serine-type endopeptidases, which are enzymes that catalyze the hydrolysis of non-terminal peptide bonds in a polypeptide chain. A serine residue acts as the active center of this enzyme. Mutations in the type 6 collagen genes are commonly associated with various forms of myopathy and muscular dystrophy. The related disease will be elaborated later. As mentioned previously, collagen is an important component of the extracellular matrix, and hence, if it is modified, it can lead to an unstable ECM that no longer attaches to cells, lowering the stability of the surrounding muscle cells and connective tissue. This leads to various diseases related to muscle weakness and contractures. This is particularly seen in aging. As presented by the data collected in this paper by Nowotny, it is highlighted that there is an increase in AGEs in older mice skin as compared to younger mice skin. This leads to the degradation of collagen. AGE accumulation in aged mice skin was detected predominantly in the deeper skin layers as the dermis and at the blood vessels, which are rich in collagen. The figure shown here illustrates the immunohistochemical staining results of mouse skin tissue, where AGEs are represented by brown coloration. Various treatments are in place to treat muscle atrophy. Conventional treatments would include exercise and physical therapy to strengthen the muscles, while more novel treatments include the use of ultrasound irradiation. From this paper by Matsumoto et al., the pulse-focused ultrasound stimulates targeted neurons to trigger signals and could prevent decreased joint mobility and collagen fibril movement. After the study was conducted for a period of 4 weeks, There was decreased muscle fiber atrophy in the muscle tissue treated with ultrasound as compared to those that were untreated with ultrasound. The protein encoded by MYH11 is a smooth muscle myosin belonging to the myosin heavy chain family. The gene product is a subunit of a hexameric protein that consists of two heavy chain subunits and two pairs of non-identical light chain subunits. It functions as a major contractile protein, converting chemical energy into mechanical energy through the hydrolysis of ATP. Myosin is found in the cytoskeleton of the cell. Glycated myosin leads to abnormal functioning of myosin and muscle contraction. Cardiovascular diseases that have been implicated due to glycated myosin include aortic aneurysm and aortic dissection. In aortic aneurysm, the aorta bulges in order to increase blood flow due to arteriosclerosis. Overstretching can cause the aorta to weaken, which can result in the bursting of the aorta when left untreated. Moreover, AGEs change the structure of myosin, preventing the normal contraction of the aorta. An open abdominal surgery must be performed to replace the damaged aorta with a synthetic graft. On the other hand, an aortic dissection occurs when there is a tear in the inner part of the aorta, leading to the separation of the inner and middle layers. Blood separates as it flows through the aorta. To correct for this condition, resection and grafting of the aorta must be performed. Otherwise, a person will suffer from high blood pressure, and the aorta might rupture if the pressure is too high. In conclusion, we have found that glycation of collagen and myosin produces AGEs that affect our muscles, which can lead to several degenerative and life-threatening diseases. Accumulation of AGEs result in the onset of degenerative disorders such as diabetes and arteriosclerosis. Maintaining a healthy lifestyle, such as eating a balanced diet, can greatly prevent the onset and progression of metabolic diseases. From this experiment, there are two hypotheses that can be investigated in the future. Firstly, we can find out whether there are any changes in the tertiary structure of glycated proteins, such as myosin and collagen, which result in their abnormal functioning. In order to do so, we can employ techniques and methods from structural biology, such as purification and crystallization of the protein, and performing X-ray diffraction or cryo-electron microscopy. Secondly, we can find out whether elevated levels of glycated myosin and collagen can be used as biomarkers for ischemic heart disease. To do so, we would analyze samples from a large population of patients with and without heart disease. Thereafter, we can perform a Western blot and quantify the amount of purified protein via BCA assay. Afterwards, we can compare whether there are significant differences in the amount of glycated myosin and collagen in patients with and without ischemic heart disease. We would like to thank Associate Professor Newman Sze, the teaching assistants, and the School of Biological Sciences from Nanyang Technological University, Singapore, for allowing us to conduct this experiment and gain valuable skills.
We have now come to the end of our presentation. We hope you have learned as much as we have. Thank you.